All right, welcome everyone to the annual WRS Student Research Showcase. I'm glad to have you here. This event is part of our WRS Visit Day for prospective students, and it's also our annual opportunity for our students to share their research. So we're going to jump right in, and we are going to start with Anna Touch. Anna, go ahead.
Hi, can everyone see my screen? Okay, let's see. All right, so my topic is on optimization of environmental DNA methods for improving detection probability of multiple aquatic invasive species in Minnesota lakes. So invasive species are a threat to the ecological and economic integrity of over 800 Minnesota lakes currently. And Minnesota lacks a standardized monitoring protocol for early detection of AIS into a lake. So something that a lot of people are now looking at is environmental DNA, or eDNA, which is organismal DNA that originates from cellular material shed by organisms into aquatic or terrestrial environments that can be detected using molecular methods. eDNA as a monitoring tool could be very effective for early detection of species across a broad geographical area. And the use of eDNA for AIS monitoring, as I said before, has become more widespread. However, it is currently not standardized, and that's what I am currently trying to do. So, like I said before, the problem is that in order to implement large-scale eDNA surveillance, eDNA surveys must be designed to maximize probability of detection of multiple AIS using eDNA. My goal is to optimize field and lab methods in order to maximize the detection of probability of multiple AIS using eDNA, and hopefully our findings and recommendations can provide guidance to Minnesota stakeholders who might consider eDNA as an AIS monitoring tool, such as the Minnesota DNR. So this past summer, I went out to 5 different lakes that have known established invasive species. So that was Pike Lake, Lake of the Woods, Vermilion, Chagua, and St. Louis River Estuary. So the 4 species that I am looking for are rusty crayfish, zebra mussel, spiny water flea, and common carp. And we know that St. Louis River has all of those, while the other 4 lakes just have zebra mussels, spiny water flea, and crayfish. The sample points that we chose was somewhat randomized by Josh Dumpke, a colleague of mine who helped me go out field sampling. And also some of these points were also sampled from the DNR. So it wasn't, we just tried to go to 10 points across the board and try to get the lake water as much as we can in order to have it like somewhat geographically equal. Test groups that we did, we collected samples in triplicates to determine the patchiness of target AIS eDNA. We did benthic water grab using a Van Doren at offshore points, and we also did a bunch of paired test groups, such as 250 milliliter grabs with a filter of 5 microns and a preserve of ethanol, 1 liter grabs with a filter of 5 microns, preserve ethanol. And then we also used a syringe kit that we could syringe out water, 60 to 250 milliliters, filter 1 micron and preserved with silica beads, and then 250 milligrams with a 1 micron filter with a C-tab preserve. I was saying that the syringe holds 60 to 250 mils is that sometimes I could not push all the water out due to the fact that there was a lot of algae blooms during the summer, so we couldn't get the 250 as we would have liked to. We also did some traditional methods such as At the nearshore surface, we did horizontal zooplankton tows, snorkeling, and shoreline searches with crayfish traps. At offshore points, we did horizontal zooplankton tows, vertical zooplankton tows, underwater cameras, and polar grabs in order to show that we actually did the traditional methods of looking for invasive species. So this is an overall method flowchart of what we are currently doing. So the eDNA collection, we've already sampled a bunch of lakes this past summer. We'll be going back to sampling this coming summer. And then we did a bunch of nearshore versus offshore and benthic. And then we did the water volume, the filter type, and the filter pore size. I'm currently at this point around here. We're using the Hydrogen DNEZ kit and the CTAPS, the chloroform method. These samples are sent to the University of Illinois to be looked at, and they will send me the data. And then right now, I'm working on conventional qPCR. And at the end, when I get all of my samples, I'll be doing digital droplet qPCR over at the University of Minnesota, Twin Cities campus. And then the overarching goal, like before, is the maximizing detection probability of multiple AAS for early detection and the prevalence of priority AAS in Minnesota lakes. So that is the end. Tried to keep it 3 to 5 minutes. Is there any questions?
Okay, now we have a couple of minutes for questions. So anyone have any questions for Anna on her research?
I had a quick question, actually. I was wondering if there's any current use of eDNA. I know you said it's not part of the Minnesota protocol. Do you know of anywhere else that uses that?
I know a couple of other projects that other students, they're working with eDNA as well. Mine is currently very methodized, so my results are my methods. And I know that the University of Illinois, they've done projects on eDNA with rusty crayfish. So we're actually in a collaborative with them and the University of Minnesota and the Twin Cities campus as well, with the seasonality portion of trying to pick the best time in order to get samples for DNA. Does that answer your question?
Yeah, it does.
Okay, perfect.
Great question. Anyone else? Okay, I didn't introduce Anna correctly at the beginning, so I will do it now, though. Anna is a first-year master's student working with Dr. Chanlin Chun in Duluth. So Anna, thank you very much.
Yeah, thank you.
Wonderful. Next, we have Ashley. Ashley is also a first-year master's student, and she is working with Dr. Jay Austin and also in Duluth. Go ahead, Ashley.
Thank you.
Are you able to remove your mask, Ashley? Are you near people?
I am on campus. So, I mean, there's no one around right now, so I'll take it off. All right. I will share my screen in a moment. It says I can't share my screen, Tracy.
Try it again, Ashley.
Says I have to leave the meeting to share my screen.
Let's see. I made you a co-host, and that didn't help. Just to.
Let's try something else.
If you want.
Okay, there we go. I think now can you see my screen?
There we go. Yep, we have yours.
Great. Okay,
Okay.
excellent. All right. Hello, everyone. Like Tracy said, I'm a first-year master's student at the Duluth campus in WRS. I'm studying physical limnology with Dr. Jay Austin. And when I'm having normal conversations and I say I'm studying physical limnology, I usually get some puzzled looks, but essentially it's just the study of the physics of a lake. So that includes water temperature, heat flux, wave structure, and other physical characteristics. To do that, we have a lot of fun. We go out in the lake and we dump some fancy buoys and other instruments into the lake. And then we leave them there for a few months. We pick them back up, download the data, and then we get some nice looking plots like you see on the right. That's just an example of one of my favorite plots that I've been able to plot with my research. And the physical limnology is important because it sets the context and the stage for the chemical and biological activity in the lake. So today I'm going to talk about an overlapping project with physical limnology and biological limnology with zooplankton. In physical limnology, like I said, we use observations over long periods of time to understand what's happening in the lake. And in Lake Superior, we have a mooring out called the Western Mooring, circled in red on that map in the top left. And it has one of the longest data sets in the Great Lakes. And I'm going to use that data from that mooring from an instrument called an ADCP, which is an acoustic Doppler current profiler, which is what this little guy is down here. And an ADCP works by sending out a sound beam into the water, and then it reflects off of particles in the water. And so we get something called a returned signal strength intensity, called RSSI. And that the ADCP is usually used to measure currents, so that calculates the velocity. But that returned, the RSSI value can tell us how much scattering or how much stuff is in the water. And so the plot on your right shows the change in that RSSI value over the course of a year. And that's what I'm narrowing in on. In oceanography, researchers correlate that RSSI value to zooplankton movement in the water by isolating a certain term, which Dr. Jay Austin has also done. And so we're taking that and applying it in limnology, which isn't a common practice. So that's part of what makes this so exciting. Like I said, I'm studying the zooplankton migration in the water. So a zooplankton, they're little tiny water creatures that make up kind of the foundation of the food web and are responsible for an important part of the carbon cycling in a lake. And this is an image of one of the species of zooplankton. There's a lot of different types of zooplankton in Lake Superior. And because we don't actually have any net tows or anything to go along with this ADCP data, we can't say anything about what species they are or specific behaviors. We just make observations. But hopefully in future projects, we'll be able to validate some of the data and characterize it more. But again, on the right, you'll see another plot. And this is the depth of that, where most zooplankton are. So where the strongest scattering signal is in the lake, That's what that plot is. And so you can see how it changes over the course of a week. But what you'll notice, which we know is established in biology, is that zooplankton have a daily migration pattern. And so at night, they go closer to the surface for feeding, and then during the day, they go down towards the bottom to avoid predation, among some other reasons. And we know that this is triggered usually by light, the intensity of light, but we don't necessarily know details about that as much. It's more of an area for further research. And with our observations with the ADCP, we noticed that during the lunar cycles, again, they were descending further into the water, or they weren't going up as far to the surface during full moons, which indicates that they're very attuned to subtle differences in light intensity, which is something that biologists can observe with net tows, but they get a small snapshot in time, versus with our ADCP, we can have a continuous data set over a really long period of time. So it makes it really interesting, and we're able to see more of those patterns clearly. And then to wrap back around to the physical aspect of limnology, what I am currently also working on is looking at this plot right here. So this is that same plot of the zooplankton depth plotted overlaid on top of the water temperature. And so what you'll notice is that during these times in the fall and spring, where the water is completely uniform, there's a descent in the zooplankton to the bottom of the water, or lower in the water column. And then in the spring, they ascend further up in the water column. And so the research that I'm going to be doing is characterizing those mixing events called convection for the physical side of it. And then hopefully that will inform us and allow us to investigate and understand the zooplankton migration in relationship to that seasonal change in the water column. So with that, I'd like to thank Dr. Jay Austin, my advisor, and Dr. Tom Hrobeck and Dr. Don Brandstrader, who provided feedback and expertise on zooplankton, and then GLERL for the funding. So with that, I'll take any questions.
Yeah, I just had a question.
Yeah.
I was curious what the light levels are that they're coming up to. Well, actually, there's no light, so they're coming up at night. But you know, at the WM station, you know, where's like the 1% light level in the water column?
So we don't have, what we're looking at with those graphs are the overnight values, and we don't have a sensor that senses moonlight intensity, just sunlight intensity. So that's something that we're going to look at for the future, because these were all historic records. This wasn't the intended data. This wasn't what we were going to do with the data originally. So hopefully maybe this summer we can put that out. But I'm trying to find ways, like with meteorological data, to kind of identify those threshold values, but I haven't done that quite yet.
We have time for one more question for Ashley. Anyone else? All right, Ashley, thank you very much.
Yes, thank you.
All right. Next, we have Nicole, and she is also a first-year master's student. She's on the Twin Cities campus working with Dr. Phelps. Go ahead, Nicole. You're on mute.
Sorry, can you hear me now?
Yep, and your slides are showing up beautifully.
Alrighty, sounds good. So this is going to kind of be a whirlwind of information, so bear with me here. But I'm going to be talking about my master's thesis, which is the effectiveness and cost of aquatic invasive species spread prevention in Minnesota. So there's currently just about 900 lakes in Minnesota that contain aquatic invasive species, and these species have huge impacts. Thankfully, there have been a lot of preventative measures to try to stop their spread. Some of these include watercraft inspection, decontamination, education and outreach, and many others. However, these different preventative measures don't come without a cost. So here's a graph showing 2020 county spending reported to the Minnesota DNR on different aquatic invasive species prevention efforts. So this data was obtained via a voluntary reporting program. So not all counties reported, but of those that did, we can see some interesting trends. On the right here are the different categories that they're spending their money on. And I think the category that stands out most to me is this one right here, this watercraft inspections category. As you can see, there's a lot of money being spent on watercraft inspections. And this black dot right here is showing the mean, the average. So what that's saying is, of those counties that reported, on average, just over $100,000 is being spent on watercraft inspections alone every year. Now that's a lot of money if you ask me, but what's even more interesting is that we don't really know how effective watercraft inspections actually are. So that's where this project comes in. We're going to fill those knowledge gaps using two different objectives, and those are the first to provide more information about how effective watercraft inspections actually are. And the second is to determine the costs of not only watercraft inspections, but other prevention techniques as well. And then ultimately, we're going to combine objectives 1 and 2 to create this holistic understanding of AIS prevention and give managers a better understanding of how to best place their prevention resources on a budget. So let's hop right into it, talking about the methods for objective 1, and that's determining the effectiveness of watercraft inspections. And the way we're going to do this is via experimental boat inspections, testing 3 levels of participants. These will be regular boaters, train level 1 inspectors, and train level 2 inspectors that will be able to use a decontamination unit. And how this is going to work is we're going to spike a boat with 3 different types of aquatic invasive species, and we're going to ask these participants to inspect and remove items from the boat as they see fit, just as they would their own boat after an outing on a lake. And a proctor is going to be there to record how much and what they remove from the boat, and that amount is going to be converted into a % efficiency. And this is going to be a standardized approach, so all participants will see the same boat spiked with the same species in the same locations. So now that we know a little bit about objective 1, let's move on to objective 2, and that is determining the cost of different prevention efforts. And for the sake of time, I'm just going to kind of briefly go over this, but we're basically going to reach out to state and county level managers via phone interviews and written surveys and ask them what they're spending on these different prevention categories. And from that data, we'll be able to get this final overarching cost that's representative of the entire state of Minnesota. So objective 1, the data from objectives 1 and 2 are ultimately going to be incorporated into this online user-friendly decision support tool called the AIS Explorer. What this does, it basically allows users to go in, and it's a virtual map that you can see what lakes are infested, which ones are at a higher risk of being infested, where you should prioritize placing watercraft inspections to be the most effective. It's a really interesting and helpful tool. I encourage you to go look at it. The URL is right down there at the bottom of the screen. So this is what the dashboard looks like right now. But when we put our effectiveness and cost data into this tool, we're going to create this new intervention impact tab. And what this is going to allow users to do is go in and create a scenario across the landscape. They're going to be able to say, I want to place a decontamination station at this lake. I want to place just an inspector at this lake. I want to place nothing at these 2 lakes. They can create whatever scenario they want. And with this tool, it's going to give them an output that will tell them the cost of that scenario and the observed change in risk. So this tool is really, really helpful in allowing managers to see this cost benefit analysis and allow them to place their resources in the most effective places to get the best bang for their buck. So we're excited for project completion in summer of 2023. And with that, I'd like to thank my advisor, Nick Phelps, the partners on the project, the MACERT graduate students, the Minnesota DNR, my funding sources. And with that, I can take any questions. And I also wanted to say that we do have some preliminary results for this project. So if you'd like to hear more about it, feel free to contact me. My contact info is right there on the screen, but thank you. I can take any questions.
Great, Nicole. Thank you. What questions does the audience have for Nicole?
Do the inspectors know they're being tested?
So the inspectors will, yes, they will know that they're being tested, but they won't know, obviously they won't know where we spiked things. So basically we're gonna go to boat launches and say, hey, would you like to participate and help us with our research? And they can either agree or not agree. And then, yeah, we're just gonna ask them to inspect this boat just as they would their own boat and see what happens. And we do realize there's some bias in that, but it's kind of a little bit inevitable. If you have any suggestions, I'm all ears.
Did you have to get IRB approval for that kind of like experiment?
Yeah, so we thought we were gonna be able to go through with the short IRB where it wasn't like based off of like, we wasn't getting any like human information, because we're not collecting information about the people, we're getting information about the general trends of how people are at, how effective people are at removing things from their boat. But we did have to go through with the longer process of IRB, and yes, it got approved already, so we're good to go. And we're expecting to roll these methods out this coming spring and summer at boat launches in both Duluth and the Twin Cities areas. So
There's a question in the chat for you, Nicole.
oh,
How do you know the effectiveness of each method used? Like the education, the watercraft inspection?
yeah, that's a really good question. So we're actually trying to kind of work out the methods for that right now. So the watercraft inspection is a little more easier to determine the effectiveness than it is for education and outreach, because there's so many different prevention or education programs, and it's hard to really conceptualize if that education is leading to action. So we're trying to figure that part out still, but the watercraft inspection is basically, I'm going to know where the things are spiked on the boat, and I'm going to be able to see how much and what they remove. And that's going to be converted into a percent efficiency. So that's how we're going to do that. Did that answer your question? Okay, Doug?
Sort of. So assuming there's going to be outreach that's going along with the effort, is it important to know the difference in time and space? Because there's been a lot that has gone on in terms of public education over the past 30 years. Sorry, I'm not asking my question very well.
Can you restate that? I might be able to answer it a little better.
Yeah, I was just thinking out loud. So there's been a lot of education that's been done in Minnesota over the past, say, 30 years. How is it important in terms of timing in which you're making the assessment? How are you going to effectively show the change?
The change in education over time.
Yeah, in behavior and education over time.
Yeah. So are you talking about as far as when we put it into the model at the end?
Right.
Yeah. So what I'm expecting is we're going to have to update these data. So right now that model is updated weekly with information. And I suspect that we'd have to continually monitor over time. Right now, we're just trying to kind of create default settings in this model that will allow users to see that cost and observe change in risk. And these are going to be default settings, but people are going to be able to go in and set their own settings. So say they got a better deal on a decontamination station unit or something, they can also change their cost and efficiency data to match what they see fit or they believe is accurate for their scenario. So they can also create those changes if they don't see our default settings to fit. But yeah, I would imagine long-term monitoring would be needed for to account for that change in in education over time.
There's also one other consideration, and that is the effort and the investment by county varies greatly across the state.
Oh yeah, that's for sure. I am going through some county data right now, and I can see the variation. And I'm still kind of going through that and trying to figure out how to, create this overarching estimate. So we're still working the kinks out of that.
Yeah, it's even going to vary among adjacent counties.
Yes, yep. And it also depends on how much each county's allocated a certain amount from the state, which depends upon how many launches they have and how much parking space they have at those launches. So how much aid they're allocated also plays into that.
It also depends upon the number of partners and how much, what the level of that partnership is by county.
Yeah, I would agree.
Perfect. Thank you, Nicole. Appreciate it.
Thank you.
Next, we have Denver. Denver is also a first-year master student, and he's here on the Twin Cities campus working with Dr. Hansen. Go ahead, Denver.
All righty. Can everybody see my screen okay?
Yes, we can.
All righty. As Tracy stated, I'm Denver Link. I am a master's student in WRS here on the Twin Cities campus, and I'm working under Dr. Gretchen Hansen. And my project focuses on the impacts of zebra mussels on walleye food webs. Just want to acknowledge some of the funding sources and partners in the projects, the University of Minnesota, MASARC, USGS, and the Department of Natural Resources. First and foremost, what are zebra mussels? Well, zebra mussels are an invasive species, an aquatic invasive species to Minnesota, native to Eastern Europe and Western Russia. They're introduced to the Great Lakes from ballast water and have spread inland to a lot of our typical Minnesota lakes. They grow rapidly in the lakes and cover hard surfaces in the lake ecosystems. And they filter algae in the lake, which ultimately compete with our native mussels as a filter feeder and have really adverse effects to a lot of the ecosystems. They clear the water, so a lot of physical characteristics change, like secchi depth and various different water aspects in that nature. And they also alter entire ecosystems. So and a lot of times they shift the energy sources from these offshore regions or the pelagic regions to the nearshore regions as they're filter feeding a lot of that phytoplankton out of the water column. They deposit those energy sources in the nearshore regions of the lake. That leads us into the second player of this story, which are the walleye. And walleye are a cultural and economically important species of Minnesota. Not every lake has walleye, but still a lot of lakes and economies depend upon the important resource that walleye are to the state. And biologically, they're constrained to a thermal optical habitat area, or a TOHA. And simply put, they like it cold and dark. And so you can really tell how zebra mussels can negatively impact walleye as zebra mussels filter feed and clear the water. And again, with climate change, walleye are going to be negatively impacted by the changes in that ecosystem. And so, like I had previously mentioned on the last slide, zebra mussels shift those offshore resources from filter feeding into more nearshore resources. And typically, walleye rely on those pelagic resources or offshore resources for their growth and survival. So what my project really tries to look at is how are walleye dealing with these changes due to the invasion of zebra mussels? Are they switching to nearshore regions for energy sources? And does that have compounding impacts? With the zebra mussels, there can be increased methyl mercury and methylation of mercury. So we're really trying to understand how does that impact public health? If they're shifting their energy source to more macroinvertebrates as young a year, and as they're shifting their impacts, how does that impact decisions and management decisions we should make for fish consumption advisory. So that was just a general overview of what the project is trying to look at. Just a quick, again, overview of some of the methods and what we do, hoping to spark some questions from you all. So in general, we're using stable isotope analysis of carbon and nitrogen to analyze how these changes are made. And simply put, different regions of the lake have different ratios of those stable isotopes. And then from there, you can compare how they are getting their energy and at what trophic level they are eating. So first, the ratio of carbon-13 shows where the consumers are eating in the lake. So are they eating offshore? Are they getting their energy from a pelagic area? Or is it more near shore, that littoral zone? And the ratio of nitrogen-15 shows at what trophic position they're eating at. In order to do this, we collect fish tissue, we collect baseline organisms, and other water parameters from the lake. So we really need to establish what is that baseline phytoplankton or zooplankton in the pelagic area. What are their baseline numbers so that we can compare the walleye and perch to, so that we can really see. And then also, again, from the littoral area, what are those macro inverts baselines so that we can compare and really tell where the walleye are pulling their energy from in that carbon source. And to do this, like I said, we collect the fish, put it in a little tube like you can see down here, and dry, grind, and encapsulate. We ultimately send our samples off to the University of California, Davis, so that it can run through a mass spec and we can get those isotope values out. And just some fun pictures here of some of the field work we did this summer. Seining is a common way that we get our fish. There's some zooplankton collection and nighttime electrofishing. So again, that was just a really broad presentation of what we are doing in the projects, and I'd be happy to answer any questions. Thank you to Dr. Gretchen Hansen, many of my field crew, undergrads in the lab helping me with a lot of lab processing, field collection, and all this such. So yeah, happy to answer any questions.
Great, thank you, Denver. We have a couple minutes for questions. Anyone? All right, thank you, Denver. Appreciate it. All right, next we have Erica Green with us. She is a doctoral candidate in Duluth, working with Dr. Kelly. Go ahead, Erica. All right, we can see your screen, but not hear you.
Sorry about that. Okay, can you hear me now?
Yes.
All right, so I'm Erica and I am a fifth year PhD student in Dr. Sam Kelly's lab up at the Large Lakes Observatory in Duluth, where we study the physics of lakes and the ocean. And my dissertation topic is on an observational study of internal waves in Lake Superior. So internal waves are sort of directly analogous to the waves that you see when you go to the beach and look at the surface of the water, except instead of operating on that like very sharp density gradient between air and the water, they operate on the density gradient that we refer to as stratification in the lake. So when the sun comes out in the summertime, warms up the top of the water, and you get this warm, less dense layer overlaying a colder, more dense layer, creating a density gradient along which waves can exist. So this is sort of a little animation of what that looks like. So this would be the warm water up here, this would be the cold water, and the yellow line is the lake surface. And a couple things I want to emphasize is internal waves, sort of counterintuitively, because their density gradient they operate on is weak, they can actually get very large. And by very large, I mean maybe tens of meters in a big lake like Lake Superior, but up to hundreds of meters in the ocean. And coincidentally, they're also can be slow. So they have periods of hours to like a couple of days. Another important thing to note is that this, even though we've got these internal waves moving along in the water, the lake surface isn't undulating. So internal waves, although they can be sort of big and active, they often have very little surface signature. So if you want to measure them, you have to go out and put instruments in the water. So why should we study these things? Why do we care about them? Internal waves move mechanical energy from their sources of energy to the water, such as wind or the tides, to the eventual sinks of mixing and turbulence. And they can move from source to sink over very long distances, certainly tens of kilometers, hundreds of kilometers, but up, thousands of kilometers out in the ocean. In addition, because all they need is a source of energy like tide and the wind, and they need stratification, they are ubiquitous. They're everywhere where there is stratification. So deep ocean, coastal shelf, and lakes, and there often can be a dominant part of the internal energy spectrum. And just to really emphasize this one point for those of you who aren't physics people, breaking internal waves vertically mix the water column. So they mix things like heat, nutrients, salt, during stratification when vertical movement is generally suppressed. So my dissertation goal is to study one particular type of internal wave called a near inertial internal wave via observation. So I'm interested in the whole life cycle, particularly the generation and dissipation aspects of it, where there's a lot of theory, but not much of it is backed up by actual observational studies. So in order to do that, we're using Lake Superior for a couple of reasons. As Dr. Austin likes to say, the lake is our lab. We're sort of using it as a lab for the coastal ocean because it's a more simple two-layer system, but it's also where we love to go out and do our work. And the way that I do it, the two main observational platforms I'm using in my thesis are Moorings. So in the summer of 2017, we went out and deployed, we had 15 mooring sites. This is like 100 kilometers from here to here, the western arm. This is Isle Royale, and this is the Apostle Islands. Duluth is like off the screen, where we deployed these moorings. They're anchored things that have a whole string of instruments you can put on them. I'm interested in temperature and velocity, so that's what was on our moorings for the most part, although we also do ship-based measurements. So we go out and deploy our own moorings on the Blue Heron, and we can also use information that the Blue Heron is gathering itself. It has a whole meteorological station where it gathers things like wind and air pressure and temperature. And then it also has hull mounted instruments. So there are things mounted in the bottom of it that measure water column velocity, for example. And then we also use profiling instruments. So here we've got our turbulence profiler that we use a powered winch to like drop off the back and pick it back up, and can in sort of almost real time measure the highly resolute temperature and shear stress in the water column that allows us to estimate turbulence. And so that's really quick what I do and how I do it. So thank you. Hello?
Thanks, Erica. Any
Oh.
questions? Sorry.
That's okay. You can always email me or Dr. Kelly also.
We have plenty of time for questions. That was really quick, Erica. Any questions for Erica?
I had a quick question. Just a curiosity. You said you kind of compared it to a wave, like breaking on a beach. And then you mentioned like breaking internal waves. Does that analogy hold, or what would be like analogous to a breaking wave that's internal? Like, what does that look like?
Well, I mean, trying to like visualize it can be a little confusing, but I mean, it's directly analogous to it. It breaks, it mixes, it creates eddies and turbulence. And, you know, changes the potential energy in the water column. So one of the things we're trying to figure out is like, what are the conditions that cause that breaking? Does it have to do with topography? Does it change with the strength of the wave? And like where and how does the energy move from maybe a windstorm on the other side of the lake? The wave travels and then it, breaks near the coast. How does that happen and what does that look like and how much mixing does that make?
This is over my head. I just thought it was
it's
interesting.
But great question. Any others?
Yeah, it's a great question. If you want to see some data, this is some actual, just very lightly processed data. So the bottom we have velocity and up here we have temperature. And so like this undulation here, the period here is like 16.5 hours. So
we can't see anything, Erica.
Oh, you can't see it? Oh, well, shoot, never mind. I don't know how to change what screen I'm sharing. Does anybody else have questions?
So maybe again, just another clarification question. So can the internal waves break in the pelagic zone as well, or is it mostly coastal? Or is it just kind of anywhere?
That's a good question. I don't really have an answer for that. I don't know what the pelagic, that's like an ocean term. They can like certainly like dissipate and interact with like mesoscale types of flows. So like big eddies and out in the ocean, or big, I don't know, other types of currents. And that is a super hot area of research, how that interaction happens, how energy is exchanged between those two kinds of flows, but it's not my specialty just yet.
Good. Anyone else? We have time for one more question. Okay, thank you, Erica. Appreciate that. All right, next we have Tesha, and she is a master's student in Duluth working with Dr. Sterner.
Hello, can everyone see my screen all right?
Yes, we can.
Awesome. All right, so as Tracy said, I am a master's student in Dr. Bob Sterner's lab over at the LLO, and I'm here to talk to you about my master's research, which involves the impact of storm-derived sediment on the formation of cyanobacterial blooms in Lake Superior. So we're going to talk about storms, phosphorus, and everything in between. So before we get to a bloom, we have to know what typically fuels it. And in terms of Lake Superior, phosphorus is the limiting factor. So that's what I want to examine. And we found a link between storms and these big cyanobacterial blooms that are happening in Lake Superior. So my research looks into whether the sediment that is reintroduced into the water column in the lake versus the sediment that flows out into the lake from the river due to erosion is more important in providing phosphorus to these cyanobacterial blooms. So the phosphorus, a lot of the phosphorus in this region of Lake Superior is bound up in the sediments, and it's inorganic phosphorus. So I decided to investigate into 3 different fractions of inorganic phosphorus. So I'm looking at total phosphorus, the bioavailable phosphorus. You can think of this as readily accessible phosphorus, and then resin extractable phosphorus, which is not readily accessible. And with the resin extractable phosphorus, I incorporated a soils method technique that uses anion exchange resins, and it's pretty similar to the like plant root uptake system that that mechanism that's in the environment. So here we have photos. This one is of one of the river sites. After, this was a couple days, or I think the day after a pretty flashy storm. And then this is a photo of Lake Superior. This is a sediment plume. So this is more likely from the rivers as well, but I just want to show a photo of the river versus the lakes. So for sample collection and analysis, I went out on Lake Superior in one of our research vessels and then collected basically lake bottom samples with a ponar. And then from those samples, took those back to the lab. And then I went and collected river sediment from within the river, along the river bank, and then like upland of the river location. So then I took those and then prepped those for the different set of analyses that I was going to run in lab. So for each of those forms of phosphorus that I mentioned, I had a different method in place. So for total phosphorus, I incorporated the ash total phosphorus method, which basically prepped the sediment and then digest it and run it through the AQ analyzer that we have in our lab. And then for the resin extractable phosphorus, we can see I'm photographed here, and those are the resin strips. And then I basically go through an entire method and then elute the phosphorus from those resin strips, and then also run that through the AQ400 analyzer. And then for the bioavailable phosphorus, I used these dual culture diffusion apparatuses that were designed with the help of Michael Plant over at the Civil Engineering Department. And basically what it is two separate chambers. And on one side, I had sediment, and on the other side, I placed algae. And then I used the growth rate of algae as a test measure to see how much bioavailable phosphorus was coming off of the sediments. So yeah, and so currently to date, there are definitely differences in the amount of phosphorus in the sediments, and basically just doing some analyses and stats right now to be able to see how significant those differences are. So I'd like to say thank you to my advisor, as well as everyone who's helped me in the lab. The list goes on and on, from lab managers to graduate students, and then also the National Park Service. Are there any questions?
I had a quick question. How exactly are you taking into consideration time or like season, in addition to like just what's going on, not necessarily weather-wise, but like any like anthropogenic activities going on prior to storms?
Yes, so for my project, I didn't really do a temporal study. So I didn't, for example, I didn't like collect sediment after a storm, or like keeping kind of like storm events. I didn't incorporate storm events into my field sampling, just because of how improbable that can kind of be. So no, I didn't take that into account. And then also, I think with the soils and sediments that we have in this region, we don't really have a lot of agriculture. So I didn't really, yeah, so basically the short answer is no, I didn't keep that in mind or use that when I was sampling.
Okay, thank you.
Good, other questions?
I had a quick question. First of all, that was amazing. But second of all, what is it you say prep these soils? What all goes into that? I'm pretty clueless about how you prep soils for that kind of thing.
Yes, so basically from the minute that I've taken it and separated into Ziploc bags, I take it back to the lab. And the first thing that I'll do is I'll homogenize the sediment, and that's just mixing it to make sure that everything that's in that sample pack is the same. And then depending on the certain analysis that I want to use, so for example, with the ash total phosphorus method, I had to make sure that those sediment samples were freeze dried. So I had to use a freeze dryer, and that just is a way of removing all of the liquid from the sample without kind of compromising the sample. So you have to, in order to do that, you have to like prep it in a 50 ml Falcon tube, freeze it, and then allow it to freeze dry for about a week. And then for the ash total phosphorus method, I had to, after freeze drying, go back and then powder or grind up the sediments and then muffle them in a furnace. And that takes like about 4 hours to do, well, depending on the tray, about like 5 samples. So the prep work takes a long time, but it's definitely like a necessity. And then for the resin extractable phosphorus, I wanted to be able to compare the resin extractable phosphorus to the ash total phosphorus. So I use the same prep method for those two. And then for the algae, I just used fresh sediment for that one.
Awesome, thank you.
Great question. Anyone else? All right, thank you.
Yep, you're welcome.
All right, next we have Bailey, who is a first-year master's student, and she is working with Dr. Cates. Bailey, go ahead.
Okay, so I think everyone can see my screen, hopefully.
Yep,
let me know
good.
if you can. Okay, great. So yeah, my name is Bailey, and I'm a first-year master's student at the Twin Cities campus, and I work with Dr. Anna Cates through the Minnesota Office of Soil Health. And just for your reference, my email contact is below. So the project title is Investigating Whether Soil Health Management Systems Improve Field Workability and Farmer Quality of Life. But I think that this is the story of rain, because water is the most destructive force on Earth. Rain has the power to shape and erode our landscapes, as you can see on the image in the right. But it's going to keep raining, and plants need water. So it's essential that the soil structure can withstand rain. It's also essential that the soil pore system can withstand rain. So this is why it is also the story of soil pores. These pores are the open space between soil structure. These spaces can be homes for microbes. These pores can store or transmit water, and these are the sites of gas exchange. As you can see in these images, pores can vary in size, shape, connectivity, and function. But most importantly, pores are dynamic and they change over time, especially with certain management practices. Tillage is a common agricultural management practice, and that changes the pore structure through the plowed soil. Plowing destroys the soil structure and the pore system, but especially those large macropores that infiltrate most of the water. In addition, rain also destroys pores. The drops will hit the surface around 60 miles per hour, and this can dislodge the particles. In the early spring, in fields that don't have cover crops, there aren't any plants growing, and so there are no leaves to intercept these water droplets. So together, tillage plus rain can destroy the surface pore systems, which then inhibits soil functioning. So these photos that I have, I took them this year, and while they aren't of the actual agricultural fields that we were sampling, it's the road next to the fields. I think that it does a pretty good job of getting at the premise of what I'm talking about, whereas having a compacted surface soil can cause a lot of damage. when they were taken right after a rain. And so rain, like I said, will destroy the surface pore system. And since the soil can't infiltrate the water because the pores were destroyed, the surface soil will remain saturated. The wet soil is a lot weaker than dry soil, so driving on these fields will cause compaction and shearing. So that means that farmers need to wait to do their field work until the soil dries. Well, in Minnesota, farmers already have extremely tight planting windows. And with climate change, there's a lot more increasing rain extremes, which is going to cause more frequent rain-related delays. These delays mean that there are less field working days, and so most likely that'll mean more stress on the farmer. So when we heard that these no-till farmers were claiming that they could get into their field a few days after a rain, whereas maybe their conventional farming neighbor had to wait a week, we decided to investigate this claim. So we have some questions that we're asking. How does agricultural management affect the way that soil structure and pore architecture changes and recovers from rainfall? And how does that affect the trafficability of these soils? Does soil health management systems change soil water behavior so that these systems have more field working days than conventional management? And how does the number of field working days affect farmer stress and quality of life? So we are going to incorporate soil measurements that I took over this past summer and next summer as well, and incorporate that in a model created by Bill Lazarus that has modeled field working days with and without soil health management systems, estimating economic benefits. We also have sent out a survey to farmers to get quality of life info. We have 20 plot trials at the Southern Research and Outreach Center in Waseca, and we have 6 farms that we take samples from. The farms are paired so that each of the 3 farm locations has a conventional and a soil health management treatment right across the road from each other. This work is fun because I get to do a lot of field work. I really love field work. I follow the weather to see when we'd get a large rain so that I could take soil samples before and after the rainfall. I use a sensor to detect the soil moisture, the volumetric water content at 6 different depths, as you can see here. This is actually an image I took this summer when the moisture tube was installed. I also get to characterize the soil at the sites. So after harvest, we got out the Giddings probe and we took some soil cores. And I had the soil judging team assist me with characterizing these soils. And that was a lot of fun. I never got to do soil judging, so it was a cool alternative. I also get to do a lot of lab work. So with those soil samples that I would collect, I would put them in water with sieves. So it's called wet aggregate sieving. And that was to understand the structure of the soil better. So I get to run other tests like running on carbon, pH, the texture analysis, organic matter separation, pore size distribution, and other assays. And I get to spend a lot of time in the soil, which is a lot of fun. I like that this work is interdisciplinary, so I get to collaborate with a lot of people, hydrologists, social scientists, soil physicists, agronomists. I get to work directly with farmers. I really enjoy that type of work. And I included my email up here. If you have any ideas or questions, I just love hearing other people's inputs. And then if you're more interested in the soil health side of things, I also have the link to the Minnesota Office of Soil Health up here. And I guess I'd like to hear from you all now if you have any questions. But one last thing, if you are curious about living in Minnesota, I've lived here my whole life, and I'm also happy to answer any questions now or email. Thank you.
Great job, Bailey. We have time for questions.
Hello, yes. I don't know a ton about soil and soil health. What is the mechanism that causes soil pores to occur in the first place? Is it biotic or something else? Or
So it's a mix, which is pretty cool. So like that one photo I showed, there was an earthworm. Like a lot of times, earthworms will kind of burrow their way into the soil. Roots of plants can can kind of make pores as well, but also freezing and thawing, wetting and drying just causes the soil structure to consolidate, and then you get the space in between the soil structure.
Good question, Rob. Who's next?
Super
Really
cool
remind
work.
me. Oh, go ahead, Tesha.
Yep. Super cool work, Bailey. I was wondering, because you said you were sampling during the summertime. Have you ever thought about like kind of what happens during like winter? Like I'm thinking of like permafrost and like those type of things, or just like the difference between like you were discussing like the freezing and thawing, or is it just more of like summertime that you were thinking of?
Yeah, cool. Okay. So I actually would say I was taking samples in the spring and summer and also in the fall. So it ended up kind of extending longer than we thought. The reason why spring was so important was mostly just because that's when the fields aren't typically covered with plants, especially if you don't have cover crops. And in Minnesota, that's when we have the most runoff. So you're most likely to get the most destruction. Like you'll see the sediment, that photo I had in the beginning, like you'll see the sediment kind of consolidate to a side just because of the water running off. But I think it would be interesting to see. Soil sampling in the winter is tough because of the frozen soil, but it's like an interesting concept. So I'd think about that a little bit more.
Awesome. Great job.
Good question. We have time for one or two more. Anyone else? Bailey, remind me, were you getting a soil science minor?
Yeah, I'm gonna get so that's pretty cool. I get to do WRS stuff, really water related things. And then I'm also getting a minor in the land and atmospheric science program. So I'll take a bunch of other soil courses. And actually, I really like that I'm doing it this way because if you are through the land atmospheric science program, maybe you would get some intro to water, but I feel like through this, soil and water, to me, they're now so interlinked that I can't imagine like not learning soil that way. So yeah, you're like WRS, that's water. How is soil related? But it's very much so related. It's a very large reservoir for the water as well.
Good. That was a good answer too, by the way. Thank you.
Yeah. So if anyone has other questions, let me know. Otherwise, send me an email. If you just want to talk about soil, let me know. I always enjoy seeing what soil looks like at other parts of the country or around the world. So my email is over here. Thank you.
Great. Thanks, Bailey. Great job. All right. Next up, we have Megan. Megan is she is a master's student in Duluth, and she's working with Dr. Brandstrader. Go ahead, Megan.
Hi guys. All right. So yeah, we're good. We can hear me. Okay, perfect. Okay, so hi, I'm Megan Corum. I'm a third-year master's student on the Duluth campus. I work with Don Brandstrader, and I want to talk a little bit about my thesis today, just like a selection of it that I think is most interesting right now. And I'm looking at potential horizontal movements of 3 planktonic predators in Island Lake Reservoir, with most of my presentation focusing on this little guy, Bythotrephes longimanus or cedar stromi, but Bythotrephes, which is the spiny water flea, and it is an invasive zooplankton. So we care about Bythotrephes because they disrupt food webs and the flow of energy from phytoplankton up into our big fish that fishermen care about, that bring in industry like tourism and are like industry specific and reliant. So in a typical food web, we have this lovely flow of energy from phytoplankton, our primary producers, through zooplankton, through fish that eat plankton, up to our fish that eat fish, our piscivorous fish. However, when Bythotrephes comes in, it eats everything it can get its little mandibles on. They are voracious consumers, and they will eat basically more than the standing stock of these prey species of zooplankton, so that they're depleting the stock. Because of that, a lot of energy is going into Bithotrepes, into these predatory zooplankton, but not a lot comes out. If you look at this picture, you can see that she has a big caudal spine, the spike on the end of her, and that is not good for fish. They will see it, but they might avoid it if they know that what it is, or they might eat it. It could either rip up their insides or stay in their gut undigested, making the fish feel full, but actually starving to death. So the energy is getting balled up in these Bithotrepes, and it can't really go anywhere. So when they get into lakes, there's a problem because the food isn't there for the big fish to get big, and then fishermen get upset. So I really wanted to look at them and their relationship to two other native zooplankton predators in Island Lake Reservoir. Bithotrepes, which I've already introduced, that big old caudal spine, was discovered in Island Lake Reservoir in 1990 and has had a sustained large population since. They're present from probably May through November, give or take, with the bulk of their population occurring in the summer. I also wanted to look at two native planktonic predators, which are Leptodora kainti and Chaeroborus punctipennis. They have some different life plans than Bithotrepes, with different predator avoidance strategies and different preferred prey types. However, they're all there in Island, and no paper has ever looked at them, the three of them in conjunction, and what they're doing and what the prey species below them might be doing. So that brings me to my question. Where are they on different scales? So the big one I wanted to look at was daytime versus nighttime, then shallow versus deep, and then across the season. So from June through October, basically. And I wanted to look at that for all three of them, plus the prominent prey species in Island, which are not, I haven't mentioned in this presentation at all, I kind of pulled them out, but I also wanted to look at how they relate to those prey species and if there are behaviors or movement patterns that relate to either each other or themselves. One big thing that's missing from this is the influence of fish. My project, I did not look at fish at all, but planktivorous fish are a huge part of this ecosystem that are essential in bringing energy up and across. There's also a lot of benthic creatures that are important too, that are also not in here, that could be influencing things. However, I wanted to look at this. So I set up a transect in the eastern basin of Island Lake Reservoir. You can see on the map there, it's about 30 minutes north of Duluth. I picked the eastern basin because it's the deeper basin. And these four stations are the ones that got sampled day and night across 2020 and 2021. Station 1 is the shallowest at 2 meters deep, and Station 4 is the deepest at 16 meters deep. And that accounts for most of the variation in lake depth in this basin, in Island Lake Reservoir in general. So basically my sampling design was the day of, we went out, we took triplicate tows of the zooplankton net, which you can see in this picture over here, that's that net by my foot, and passed them into jars. And then we went out an hour after sunset and repeated it and caught the nighttime zooplankton. The interesting thing about Island Lake is that it is a stained lake, so the water is very dark brown, which should be a hindrance to Bithotrepes. They have this big eye spot and they are visual predators. So you would think that they would not do well here in this stained lake, but they do for some reason. So we went out and we sampled this for 2020 and 2021. So I'm going to show, I've pulled out what I think are two of the most interesting graphs right now of my Bithotrepes data, because there's a lot of data. But I want to show this one. So this is a percentage graph of comparing percent distribution of Bithotrepes in 2020 across the four states for the months. So the top is August, the August sampling date, day and night, then September, day and night, October, day and night. Remember, station 1 is the shallowest at 2 meters. Station 4 is the deepest at 16 meters. The most interesting pattern to come from the 2020 sampling was this October nighttime. Huge percent of the population is showing up at that station 1, that shallow station in October nighttime. And I didn't really have a good explanation for it. I wanted to see if we could repeat that pattern this summer and see, is there something there? Like, are there a bunch of them? But that is the big pattern, plus also the higher number of animals caught during the nighttime than the daytime for across all samples. Okay, so that's 2020. Same graphs for 2021, adding June and July up at the top. In 2020, we were not able to sample June and July because of COVID protocols, but we could for 2021. So those got added in, and those showed some pretty remarkable numbers of animals, especially that July nighttime, going from 1,400 animals across the transect in the replicates in the day to over 10,000 animals at night. That's a huge difference, and they have to be coming from somewhere, which is kind of the question I was asking, where are they coming from? The sad part here is that we were not able to capture the same trend in October. Conditions for the water were very similar. The time of year was similar. I think the moon was a little bit different, but you can't control the moon. But you can see that there was not that huge density of animals at that October very last graph on there. But there was a shift where there's a higher percentage of animals at station 2, the second, like the medium station during the nighttime and the daytime. I don't know if that means anything. So those are just distribution patterns of one of the 3 main animals in my project. And really on their own, they really show that, well, there's some patterns there. There's some interesting things happening, especially with this if you look at day night for 1-to-1. But on their own, they don't really tell me much. So like going forward, I'm still working with the K. arboris and the Leptodora data, so the other predators, and then all of the prey data is also being processed right now. Lots of analysis. So are they moving horizontally? Maybe, but I can't tell you for sure yet. Does anyone have any questions?
Great job, Megan. Questions for Megan? You were too thorough, Megan, sorry.
I guess. I even cut stuff out. I have so many graphs.
I had a quick question, actually. What does a prey for these guys look like? What are they eating?
Yeah, so the thing they like the best is Daphnia, which is, a lot of people have heard of them as like the big plankton that's everywhere. Daphnia is the big one. So any kind of cladocerans or copepods, and those guys eat algae mainly. They're filter feeders that are just grazing all day long. But Daphnia are the big ones. So most of my prey graphs, I'm focusing on how the Daphnia are moving and their biomass, because they're what really matters.
Sweet, thank you.
Good question. Anyone else? All right, thank you, Megan. Appreciate it.
Thanks, guys. Let's see if I can figure out how to stop this.
All right, up next, we have Victor. Victor is a doctoral candidate on the Twin Cities campus, working with Dr. Niebuhr. Go ahead, Victor. If you can, take your mask off.
Oh, I can't. Can you hear me?
Yes, we can hear you.
Okay, perfect. Can you see my screen?
Yes, we see it now.
Perfect. Okay. Thank you, Tracy. Hello, the prospect grad student of the WRS program. My name is Victor, the PhD candidate. The PhD candidate of the program, and I would like to briefly present a simplified version of my result. Before my presentation, I would like to ask, why does the water storage matter? Water storage matters because flooding matters. Flooding is one of the most devastated natural disasters in the United States and worldwide, both financially and socially. However, a good flood prediction should not only include the rainfall and the characteristic of the watershed, but also the spatial distribution of the water storage within a watershed. Studying storage discharge relation plays a significant role in the flood prediction. To determine how the water storage can improve the prediction of the discharge, I would like to answer two questions. First, can we improve the stream flow prediction by including the total water storage within the boundary of the watershed? Second, can the downscale water storage improve the stream flow at a more regional level? In order to answer those questions, I use the hydrological model at the Rhone River watershed, shown on the map on the right side of the slideshow. This diagram briefly summarizes my research that can answer my first question. In my control group, I will test the simulation with 7 weather variables and the total stream flow of the watershed. And in my experimental group, I will not only include the I will simulate the stream flow with not only only 7 variables from the control group, but also the different layers of the water storage. So that my control group will simulate one set of the stream flow prediction that exclude the water storage. And my experimental group will simulate 7 sets of the stream flow prediction that was based on including different layer of the water storage. The result will be shown with a comparison between the simulated stream flow, including and excluding water storage. And you will later will see the bar chart of the result that with a coefficient scale from 0 to 1, when 0 is the lowest and 1 is the highest. In the second part, to answer my second part of the question, this diagram will answer the second part. In the control group of the second part, I will simulate the local stream flow that with the 7 variable at each of the sub-watershed, 131 watersheds within the Rhone River watershed. And my experimental group, I will simulate the 131 local stream flow with both the weather input and the local water storage that has been downscaled. And the control group will simulate 131 stream flow prediction, and the experiment group will simulate 7 stream flow prediction of the 131 sub-watershed within the boundary of the Rhone River watershed. And here is the first result. When the blue hydrograph is the stream flow of the observed data, the red result is a simulation without storage. On the left, on the right, the orange hydrograph is the result of the simulated stream flow with the water storage. And you can see the improvement by including water storage. And this bar chart shows the improvement of the simulation by including water storage. And you can see that by including total water storage, it can improve. But by including active groundwater storage, it can improve the best. And for the local watershed, the result shows that by including the downscale result of the total water storage, the stream flow prediction will be improved, showing in the green area. And with the active groundwater storage, interflow storage, and surface storage, you can have the both best improvement for predicting the local stream flow. And this box chart shows that by including the active groundwater, they have the 0131 watershed have the best improvement of predicting the local stream flow. So overall, in conclusion, by including the total water storage, it can improve the total stream flow prediction at Round River watershed. By using a downscale water storage, we can improve the prediction of the stream flow at a local level. And among different layers of water storage, the active groundwater can improve the stream flow the best. Thank you. I will use this time to thank you, my advisor, Dr. Niebuhr. I will also thank Tracy for this opportunity, as well as the BBE and my research team, which have helped me for all the works. Thank you.
Thank you, Victor. Any questions for Victor?
Tracy, I've got a question for Victor. I'm wondering, it surprises me to hear about these, that you're adding storage to flood predictions, because I would assume that would be a common practice. So what is the common practice for making flood predictions? Does it just not account for storage typically, or just not account for it as well as you
That's
are?
a very good question. Typically we don't in the more right now we use a lot of hydrological models, and right now the National Weather Service use the build a flood prediction model based on the reservoir flow, snow water content, river, and the reading from the rain gauge and river gauge. So water storage is a difficult subject because it is not easy for you to just monitor it. So yeah, it was not widely popular. Even right now people try to use the machine learning algorithm to predict rainfall, and they found out that they haven't really used water storage a lot either. So no, they're not widely used right now.
Thanks.
Oh, I will start sharing.
Any other questions for Victor? All right, thank you, Victor. Appreciate it.
Thank you.
All right, next we have Molly. And Molly is a second-year PhD student working with Dr. Johnson and Dr. Hoffman in Duluth. Go ahead, Molly.
Hey everyone, thanks Tracy. I will start by saying, yeah, I'm working with Joel Hoffman and Lucinda Johnson, and I'm supported by a fellowship at the Lake Superior National Estuarine Research Reserve up here in, well, it's actually in Superior, but I'm in Duluth. So the focus of my research is called cultural ecosystem services in the St. Louis River estuary. So the US has spent over $87 million on cleaning up the St. Louis River estuary since 2010. This has been done under the Area of Concern program that is happening across the Great Lakes to address the loss of beneficial uses of the estuary. And it includes remediation, restoration, and some components of revitalization. So for those of you that aren't from this area, the St. Louis River is the estuary, is between the communities of Duluth and Superior. And my study area is the direct watershed that flows into the estuary and the Fond du Lac Reservation as well, which is connected traditionally with the estuary. So the catch is that we've made this huge investment, but we don't really know how the community has benefited from that. For the type of projects being done, we typically measure environmental targets like sediment quality, water quality, wildlife populations, those sorts of things, because that's what those projects are designed to address. And we just sort of assume that with a healthier environment, the community will benefit, but we don't usually measure those benefits. And we also don't know if people benefit equitably because we also don't measure that. So to ensure that these kinds of investments really do benefit the community and equitably, we need some sort of supporting data to be able to evaluate that. And that is where cultural ecosystem services can come in. So cultural ecosystem services are the intangible benefits that people experience from nature. And they're maybe the most direct way that people on an everyday basis benefit from healthy ecosystems. So that includes sense of place, cultural traditions and heritage. This is a photo of wild rice. The social relationships that develop around the water, aesthetics and inspiration that come from the water, knowledge systems and education, recreation and tourism, and spiritual and religious values. Am I sharing my screen?
No, Molly, you're not sharing your screen.
Dang it.
You were going so wonderfully that
So
I just thought you were
fast.
going to go without a presentation.
Oh God, okay, can you see it now?
Yes, recreation and tourism.
Okay, well, I'm sorry, I had
Well,
great
you
slides.
did a
but
great job. Keep going.
I'm just gonna keep going. Anyway, and spiritual and religious values as well so these are all things that are associated with cultural ecosystem services. So my research questions for my dissertation are focused on understanding how cultural ecosystem service experiences are distributed across the study area, and then what major things influence cultural ecosystem services. And that includes how personal identity influences cultural ecosystem services. And by personal identity, I mean both demographics, sort of what socioeconomic group people fall in, as well as personal life history, those like personal experiences that make everyone unique, how those things influence cultural ecosystem services, and then how the landscape influences cultural ecosystem services. And for this study, I'm focusing on how access to the water, how the amenities at the water, and how the ecological condition influences cultural ecosystem services. So this is really important as we start to or continue to adapt to a changing climate, because we know that the impacts of climate change will be hardest on vulnerable and underrepresented communities. So overall, my goal is to be able to identify management interventions to ensure that our investments in the river have the most benefits for the most people. So my project is based on 3 different big parts. And the first one is called the Waterway Benefits Survey. And this will be a quantitative survey that asks people questions about their experiences with and benefits from the river and the water. And it'll be focused on a convenience sample targeted towards diverse age, race, and income groups. And my goal is to get 500 responses. The survey will be followed up with semi-structured interviews And those will be more qualitative and have more in-depth questioning about people's experiences with the water, their barriers to experiencing cultural ecosystem services, and how those experiences and barriers connect to who they are as a person or personal identity. That'll be based on an established protocol, and my goal will be somewhere between 30 and 50 interviews. And then lastly, I'm going to collect and compile public data on landscape factors, including the locations where people can access the water, what amenities are there to facilitate that access, and the condition of the ecosystems. So it's a mixed methods approach, and so my data analysis will reflect that as well. And I will analyze the survey data more quantitatively and test for correlations between cultural ecosystem service experiences and demographics, and cultural ecosystem services and landscape factors. And then to sort of better understand those trends that I am able to identify in the quantitative data, I will use a qualitative approach to analyze the interview data. And that'll involve identifying themes in the interviews and looking deeper into how different intersectional groups have different cultural ecosystem service experiences. So recognizing I'm in my second year and just sort of beginning to start to collect data, I just thought I'd talk a little bit about the process, because studying social ecological systems is pretty different from focusing on the ecology itself. And so my study design has been informed by 2 different advisory groups, an Indigenous advisory group to help make sure that my survey methods and interview methods are able to capture cultural ecosystem services associated with Native Americans in the study area. And then I have gone through institutional review board approval through the university, and I'm also going through the Fond du Lac tribe has their own institutional review board, so I am working through that process right now. Hope to begin to collect data, and then because this project is engaging the community so much, I think it's really important to be able to share results. And I'm lucky to be a fellow at the Lake Superior Reserve and be able to use that as a means to get the data out there and the results out there, and be able to share the input of the participants in the community back with them through some sort of public display, as well as making recommendations available to resource managers. So I see this as sort of an ongoing collaboration with both the tribe and those advisory groups that have provided input. And sorry about messing up my slides, but if you have any questions, feel free to reach out, and I'm happy to answer any questions.
You did a good job, Molly. You don't need to apologize. Great, thank you for that. Anyone have any questions for Molly? See, you did so well, they don't have questions. Good job. Thank you very much. And our final presentation will be from Mariel. Mariel is a PhD student on the Twin Cities campus. Take it away.
For sure. I am the last one. So after this, everyone gets to take a break. So let's see here. All right, I'm assuming, hopefully everyone can only see the presentation. Okay, excellent.
Looks good.
Perfect. All right. So like Tracy said, my name is Mariel. I'm a second-year PhD student working with Dr. Shui Feng, who works at SAFL as part of the Civil Environmental and Geoengineering Department. So I'm going to talk to you guys today a little bit about some of the recent results from my ongoing research, which focuses on streamflow generation processes in peatland systems, specifically the peatland systems at the Marcel Experimental Forest, which is this really cool site about 45 minutes north of Grand Rapids. And it's part of the Experimental Forest Network that's run by the USDA and USFS. And it focuses on studying watershed processes through paired watershed studies. So it has kind of a truly remarkable amount of interesting data relating to hydrology, climatology, land cover, various cool things. And so this project actually started when we noticed that there was this very clear trend in decreasing streamflow in both the S2 and S5 watersheds, which are the two control watersheds at Marcel. And we were interested in what might have been causing this decline. So normally when you see a decrease in streamflow, especially in a place like Minnesota, where winter takes up half of the year, the immediate instinct is that there must be something changing in the snow season that's causing this. But when you actually look more closely at the climate data at Marcel, there's not only been no change in snowfall, but there's actually been no change in precipitation or temperature at all. So it kind of further increases this question of what exactly is going on here. But there has been a consistent decrease in water table elevation, which kind of signals to us that there's likely been something happening within the watershed between the time that the water is entering as precipitation and leaving as streamflow that's causing these changes. So to really be able to look into this question, we utilized as much of the Marcel data as we could to try and determine kind of the clear flow path that the water was taking from snowpack to stream. So to do this, we kind of added all of the data together and decided to determine several dates of important hydrological events, like the date of peak snow or the kind of like the date of the water table peaking, or like the date that the streamflow onsets. And so you can see here those 6 indicators that we pulled out for our sample year of 2016. And you can see that in this year, they come in a certain order. But as this is a sample year, we kind of wanted to know whether this was just 2016 or whether this was the kind of common path that the water took. So we repeated this process for all 25 years of data. And it does turn out actually that this is the average path that kind of water is taking through our watersheds. Which is really interesting because it illustrates this kind of fill and spill process that's happening, where the water first accumulates in the snowpack, and then as the snow starts melting, it then percolates into the water table or runs off over the surface. And then once the water table has kind of exceeded the surface level, then there's this kind of large influx of stream flow, and you usually see that stream flow peak shortly after. So while this ordering is really interesting, it doesn't quite get at our question of what is causing the streamflow decline, but it does show that there's this really clear connectivity across the landscape. So to really dig into the question of what the internal processes affecting streamflow at Marcel are, we turned to some statistical modeling. So we did this by generating a model to predict streamflow using all of the interconnected variables from before. So specifically, we're using a technique here that is called stepwise regression. So meaning we start from the full model that's here at the top of the screen, which contains all of the variables of interest. And we essentially generate a lot of models using every combination of subset of terms from this full model, and then we relate them. I know that explanation is a little bit confusing and doesn't really talk at length about what's going on here. So if you'd like to chat more about that, I'd be happy to, but for the sake of time, I'm gonna leave it at that. The important takeaway is essentially that this technique allows us to get at how important each of these elements, snow, water table, frost, temperature, et cetera, are in determining stream flow. So think of it kind of like if we were to find like a perfect model, which we're not going to, but if we were, the likelihood that any one of these watershed components would be in that model is the likelihood that you see here in this bar plot. So the super complicated kind of interaction terms that really don't have any physical meaning in the watershed aren't very important. Their weights are really small. But the individual terms are very important in dictating the amount of stream flow out of our catchment. And what's the most surprising here is actually this one right here at the bottom, which MFT stands for mean frost thickness. So this is frost actually, which is coming out as the most important variable. And I should mention that, like, if you were to go out to Marcel and only measure frost and expect that to be a good indicator of stream flow, you're probably going to be sorely mistaken. It's not going to work great. What this more accurately shows is that frost is a really good companion predictor, meaning that if you add frost to an already decent model using precipitation or water table elevation, your ability to predict stream flow is going to increase significantly. So what does this all mean kind of in the large scheme of things? It means that frost essentially plays a larger role in dictating the amount of stream flow than we originally thought. So our next steps, which are still a little bit more hypothetical at the moment, this is an ongoing project, are to look more closely as to how this kind of frost affects the partition between precipitation and evapotranspiration here in Minnesota. So there's a lot of really interesting research currently looking at the influence of climate change on this partitioning process. For instance, there's a really cool graphic here showing from a recent paper that's looking at how the both amount and distribution of snow is affecting this partition. But you'll notice that even though they're considering snow, they actually don't consider frost at all in this paper. And so while we don't exactly know how frost will affect these relationships, it's kind of a fun new horizon, and we expect it to kind of show some interesting secrets. So thank you for listening. If you have any questions, feel free to ask or shoot me an email.
Thanks, Mario. What questions do you all have for her? Yeah, good job with your presentation, Mario. Thank you.
Thank you.
Any questions? All right, well, thank you. And thank you to all of the students who presented today.